introduction to the nervous system here with last couple of sections of the chapter. So we're talked about neurons themselves. We've talked about the way that neurons interact with each other. Now let's talk about the language they use to speak with each other, and that is the neurotransmitters. There are at least 50 neurotransmitters, and I would suspect that's a low estimate for the number of neurotransmitters. Most neurons are going to make two or more neurotransmitters, so they can exert several different influences. The choice between which of those transmitters is released is going to depend on things like stimulation frequency. But in general, neurotransmitters are classified either by their chemical structure or by their function, sometimes both. So we can start with the one that was first identified and best understood, the one that's released at the neuromuscular junction, and that is acetylcholine. Acetylcholine is also going to be used by the autonomic nervous system and some neurons within the central nervous system. It's synthesized from a molecule called acetic acid and a choline molecule, and an enzyme called choline acetyltransferase builds acetylcholine. And if you remember to the muscles, we talked about this then too, it's degraded by an enzyme called acetylcholinesterase. There are a bunch of neurotransmitters that are classified as biogenic amines. Catecholamines would include dopamine, which we've heard of before, norepinephrine we've heard of before, epinephrine we've heard of before. Those are all derivatives from one of the amino acids, one of the 20 common amino acids called tyrosines. The other type is the indolamines, and serotonin, you've probably heard of, is made from the amino acid tryptophan. Histamine, we've talked extensively about as an inflammatory chemical. It is also a neurotransmitter. It's made from the amino acid histidine. These are all widely used in the brain. They play a role in emotional behaviors. They play roles in biological clocks. Some of these are used by the autonomic nervous system motor neurons, norepinephrine in particular. And when we get to the autonomic nervous system, we'll see that in more detail. Imbalances in these are associated with mental illnesses. It's thought that a lot of the drugs that treat mental illnesses like depression are affecting these neurotransmitters, the balances of these neurotransmitters. Amino acids make up all proteins. There's 20 of them. Difficult to prove which are neurotransmitters, but there's some that we are very sure that have been studied quite well. Glutamate, aspartate, glycine, and GABA, which stands for gamma aminobutyric acid, which is why we call it GABA. Short peptides, strings of amino acids that have diverse functions. Substance P is a mediator of pain. The endorphins, beta endorphin, dynorphin, and enkephalins are natural opiates. They help reduce pain perception. There are some gut-brain peptides, somatostatin, cholecytokine, play a role in regulating digestion. We'll see more of those when we talk about the digestive system. Purines, one of the two types of molecules that make up DNA and RNA. They are nucleic acids used in the central nervous system and peripheral nervous system. ATP, we always talk about it as an energy molecule. It is also a neurotransmitter, and it has other effects that we'll see through the course of the year. Adenosine is an inhibitor in the brain. Caffeine blocks adenosine receptors. Purines can also induce calcium influx in astrocytes, the glial cells. Some gases and fats are also neurotransmitters: nitric oxide, carbon monoxide, hydrogen sulfide. They bind with G protein coupled receptors in the brain. Lipid soluble, so they can go through membranes. They can be synthesized quickly on demand using enzymes. Nitric oxide is involved in learning, the formation of new memories, as well as brain causing or linked to brain damage in stroke patients and smooth muscle relaxation in the intestines. Hydrogen sulfide acts directly on ion channels and alters their function. Endocannabinoids act on the same receptor as THC, which is the active ingredient in marijuana, the most common G protein linked receptor in the brain. It is lipid soluble, again synthesized on demand, thought to be involved in learning and memory, and may be involved in neural development, controlling appetite, and suppressing nausea. Functionally, the neurotransmitters, those neurotransmitters we just went over by their structural classification, can be classified either by their effects or by the actions they have. Excitatory versus inhibitory. So neurotransmitters can have an excitatory, they can help to depolarize a cell, or inhibitory, they can help to hyperpolarize a cell. And that effect is going to be determined by the receptor on which it binds. So GABA and glycine bind to inhibitory receptors. Glutamate usually binds to an excitatory receptor. Acetylcholine and norepinephrine have at least 2 receptor types, and those receptor types have opposite effects. So acetylcholine is excitatory at the neuromuscular junction in skeletal muscle. We said that all along. But acetylcholine is inhibitory on cardiac muscle because the receptor on cardiac muscle, the acetylcholine receptor, is different than it is on skeletal muscle. Direct versus indirect actions. Direct action, the neurotransmitter binds directly to and opens an ion channel. That gives you a rapid response, altering membrane potential. Acetylcholine did that. Most of the amino acids work that way. Indirect action, the neurotransmitter is going to act through an intracellular second messenger system. And that second messenger system is usually a G protein pathway. We've looked at these before. We'll review it again in a moment. Those indirect actions, that intracellular second messenger system gives you a longer lasting amplified effect. Again, one receptor is bound, but many second messengers are created. So the biogenic amines, the neuropeptides, and most dissolved gases are going to work through the G protein pathways. They can be neuromodulators, chemical messengers that are released by neurons that don't directly cause EPSPs or IPSPs, but instead have an effect on the strength of the synaptic transmission. So they might influence the synthesis, release, degradation, or reuptake of a neurotransmitter. They might alter the sensitivity of the postsynaptic membrane to the neurotransmitter, changing receptors on it. And they may be thought of as a paracrine. They're acting locally. Channel length receptors, they have ligand-gated ion channels. Their action is immediate and brief. One ligand, one receptor, one response. Their excitatory receptors are channels for small cations. Sodium influx contributes most to depolarization. Inhibitory receptors would allow calcium influx that caused hyperpolarization. Sorry, I said calcium, it's chlorine, a negative charge, and negative charges make it more negative inside. G protein linked receptors, the responses are indirect, much more complex, slower, but the trade-off for that is a prolonged response. It's going to involve a transmembrane protein complex. It's going to cause widespread metabolic changes within the target cell. Its second messengers amplify the responses. And some of the examples of these are the muscarinic acetylcholine receptors, receptors that bind biogenic amines, and receptors that bind neuropeptides. The mechanism for this to work is the neurotransmitter binds to the G protein linked receptor, activating a G protein. That activated G protein controls the production of second messengers. Those second messengers can be things like cyclic AMP, cyclic GMP, diacylglycerol, or calcium. And those second messengers are going to have widespread effects on the cell, including opening or closing ion channels, activating kinase enzymes, again, kinase enzymes phosphorylate other enzymes, turning them on or off, They can phosphorylate channel proteins and even activate genes causing protein synthesis. So we have seen these before, but just to quickly review, we have a neurotransmitter that serves as the first messenger. It is binding to a G protein. This is a G protein. This is a receptor. They are coupled together. When that neurotransmitter binds to a G protein coupled receptor, GDP is removed from the G protein, GTP is added to it, and that activates the G protein. That G protein will go find a second target, and in this case it's going to be adenylate cyclase. The adenylate cyclase enzyme takes ATP and converts it to a cyclic AMP, takes two phosphates off and folds the two ends together to give you a circular molecule. And that cyclic AMP acts as a second messenger. It changes the membrane permeability, opening and closing ion channels. It also activates enzymes, and it can turn on specific genes. So we get a widespread response amplified because even though it was only one neurotransmitter, there's a whole lot of cyclic AMP made in the cells. Okay, so how does this information get integrated? How does our brain make sense out of the world? Well, if I told you I completely understood that, I would be lying to you. If I told you anyone completely understood that, I'd be lying to you. But we do know some basic ways that information is processed, integrated, neurons working together in groups. Those groups are going to contribute to the broader neural functions. There are billions of neurons in the central nervous system. They're all hooked up to at least thousands of other neurons, and we must have integration so that the parts fuse to make a smooth operating whole. But in reality, most of it is still a big black box to us. Let's start with organization of the neurons. There are different ways that neurons can be pooled together. Functional groups of neurons can integrate incoming information received from receptors or other neuronal pools and forward that processed information into other destinations for continued processing. We can have a simple neuron pool, which is single presynaptic fiber branches synapses with several neurons in the pool. In that case, we would have a discharge zone. Those would be the neurons close to the incoming fiber, and they are more likely to generate an impulse. A facilitated zone, neurons on the periphery of that pool, further away from the fiber, usually not excited unless a threshold or stimulated by another source. So here is our input. This is our discharge zone. These are facilitated zones. Usually the signals will go back through here, but if we had learning involved, we might turn the whole thing into a discharge zone. Processing, we can process it in a couple of different ways. We do serial processing. Input travels one pathway to a specific destination. One neuron stimulates the next one, which stimulates the next one, and so on. This works in an all or nothing manner, produces a specific anticipated response. Serial processing is going to be seen when we look at spinal reflexes. If you use a hammer and strike the patella tendon, you will get a response because of serial processing. These are rapid automatic responses to stimuli. A particular stimulus will always cause the same response, and it will occur over a pathway, and we're going to call that pathway a reflex arc. And that reflex arc, that pathway, is going to involve a receptor, a sensory neuron, a central nervous system integration center, a motor neuron, and an effector. So a stimulus comes into the receptor. That receptor triggers a sensory neuron. That sensory neuron sends it to an integration center. In this case, it's in the spinal cord. It's consisting of a single interneuron. That interneuron signals a motor neuron, which sends a signal to a skeletal muscle in this case. So we have a simple reflex arc. We can also have parallel processing, in which input will travel along several different pathways. Different parts of the circuitry deal simultaneously with the information. One stimulus is going to produce numerous responses. This is very important for higher level mental functioning. A scent smell might remind you of an odor and an associated memory that goes with that odor. Scents are very good triggers of memory, most likely due to parallel processing. We also have circuits. Circuits are patterns of connections within neural pools, and there are 5 types of circuits: diverging, converging, reverberating, and parallel after discharge. So in a diverging circuit, we get one input. That input is passed along, and the information spreads out, and we get many inputs after that. It could be, and it is an amplifying circuit. Converging circuits, several different circuits converge on a single output. Different sensory stimuli can elicit the same memory, so you remember something when you smell, you see a picture of it, or you see a picture and you hear a sound. All of that goes to a single memory. Reverberating circuits oscillate back and forth to each other. Signal comes in, is passed along, but eventually it makes its way back. That leads to an output. Sleeping, wake cycles, breathing rhythms, repetitive motor activities involve these reverberating circuits. Parallel after discharge circuits means you hear a single stimulates neurons arranged in parallel arrays that eventually converge, so coming in, diverging, but converging again to a single output. So mathematical calculations, here's your, if you will, your math problem. You go through all kinds of different permutations, but you end up with a single output. More than one way to solve the problem. Developmentally, the nervous system originates from the neural tube and neural crest cells that are formed from the ectoderm. That neural tube is going to become the central nervous system. The neuroepithelial cells of the neural tube proliferate into cells needed for development. Neuroblasts become amitotic, begin to migrate, and neuroblasts will sprout axons and make connections with targets and become neurons. Location for that to begin to occur is called the growth cone. It's a prickly looking structure at the tip of an axon that allows it to interact with its environment, and it does so with cell surface adhesion molecules like laminin, integrin, nerve cell adhesion molecules, also called NCAMs. They provide anchor points. There are neurotropins that attract or repel the growth cone. One of the factors that works with them is nerve growth factor that helps to keep that neuroblast alive. And there are filopodia on the growth cone that follow the signals towards the target, doing a positive chemotaxis, hoping to make connections. Once that axon finds its target, it has to find the place to form the synapse. Astrocytes help with that. Cholesterol is needed for membrane construction and synapse construction. About 2/3 of our neurons die before birth. If an axon doesn't form a synapse with its target, they're triggered to undergo apoptosis. And many other cells also undergo apoptosis during development. During childhood and adolescence, learning is going to reinforce certain synapses, and it's going to prune away others. Neurons are amitotic after birth. There are some exceptions. We'll see those when we talk about the olfactory system and the hippocampus. Here's an image of that neural growth cone, filopodia coming out, being directed to where it's going, and hopefully it will make a connection. Hopefully that connection for you will be related to anatomy and physiology, and you'll be able to fire this neuron to give you fire the right neurons when it comes time to take a test.